Hello friends, welcome to With My Plant channel. Today we are going to understand about the biosynthesis of cytokinin. We are in cytokinin series. In the previous class we understood about the discovery of cytokinin. So let's start the biosynthesis. The first step in biosynthesis of isopentenyl cytokinin is N-phenylation of your AMP, ADP or ATP, anything can be, with your DMAPP and HMBDP. DMAPP full form is dimethyl allyl diphosphate and HMBDP is hydroxymethyl butynyl diphosphate, which is catalyzed by your adenosine phosphate isopentenyl transferase. This is an important enzyme which is initiating the biosynthesis of cytokinin, that is isopentenyl transferase, or otherwise you can call it as IPT. Hydroxymethyl butynyl diphosphate basically is intermediate of one secondary metabolite biosynthesis pathway, that is methyl erythritol phosphate pathway, that occurs in bacteria and plastid, which means it is happening in your prokaryotic cells. A second chemical we read is dimethyl allyl diphosphate, that is DMAPP, is synthesized basically via both the pathway, that is the MEP pathway, and other one is your mevalonic acid pathway. So, and it is very common in your eukaryotic cells. So question may come that which pathway is responsible for your biosynthesis of cytokinin. If it is in particular with eukaryotes, you can say both MEP and mevalonic acid pathway, and if it is termed as bacteria, in bacteria or plastid or any prokaryotic organism, then we can say it as it is because of this MEP pathway. Okay, so let's understand the steps here. What are the steps to biosynthesis of cytokinin? Here are the steps. I have divided whole the process in 1, 2, 3, 4, 5, 6, 7 steps. Okay. So the first step is ATP, or ATP is the one who is going to start the process of biosynthesis of cytokinin. So ATP or ADP, you can say ATP or ADP, adenosine triphosphate or adenosine diphosphate, is converted to IPT, iPTP or iPDP, or otherwise called as isopentenyl adenine riboside triphosphate or isopentenyl adenine riboside diphosphate. Otherwise, it is called as iPRTP or iPRDP. Okay. Third is your ZTP or ZTP, zeatin riboside triphosphate or zeatin riboside diphosphate. Here this riboside group is being removed to form your zeatin, phosphate group is being removed to form zeatin riboside, and again this riboside group is being removed and to form trans-zeatin. So this is the bioactive zeatin. The structure is like this, and the inactive or inactive state form of this zeatin is basically O-glucosyl trans-zeatin. So cytokinin is synthesized in roots. This is a very important question, that where the kinin is being synthesized, and it is basically the roots, where they are transported to shoots through xylem. The biosynthesis of purine type cytokinin is more clearly known than other types. Various components of purines are derived from molecules like formyl tetrahydrofolate, aspartate, and your carbon dioxide, glutamine, glycine, methyl, uh, diene tetrahydrofolate. So the biosynthesis of purine nucleotide is basically, yes, this is important. What is purine nucleotide? Purine nucleotide is basically your purine base, ribose sugar, and phosphate. So in the previous slide also we understood that adenine ribose phosphate is being formed, and in the first step this phosphate is being removed, and second step ribose sugar is being removed, and this adenine is converted to your cytokine. So this in the first step, as we read in the previous slide, the first nucleotide to be synthesized is basically inosine monophosphate, which is forming your AMP. Okay, inosine monophosphate converted to either guanosine monophosphate or your adenosine monophosphate. Free cytokinins are synthesized from adenosine monophosphate and isopentenyl pyrophosphate by condensation reaction in the presence of enzyme isopentenyl transferase, otherwise called as IPT. The product formed is N6 delta 2 isopentenyl adenosine 5 monophosphate, and the compound is supposed to be precursor of all cytokinin. Okay, so this is the chemical, that is N6 delta 2 isopentyl adenosine 5 monophosphate. So this compound is converted to isopentenyl adenosine by removing the phosphate group, and further it is converted to your N6 isopentenyl adenine by removing the ribose sugar group and hydroxylated to form your free zeatin. So the reduction of the double bond in isopentenyl side chain of zeatin would give rise to your dehydrozeatin, which is again a inactive cytokine. So this is the whole process of biosynthesis of cytokinin. So here, as I mentioned the steps, it is easy to remember all the compounds in the whole process of biosynthesis of cytokinin. So this is all about today's class. If you like this or if you understood everything, then please press like button and share and subscribe this channel and this video. Thank you.